today Dr. Sonya MacParland. Dr. MacParland is a research scientist at Toronto General Hospital Research Institute, and she is assistant professor, Department of Laboratory Science and Pathobiology and Immunology at the University of Toronto. Dr. MacParland's research program is focused on translating fundamental knowledge about the immune biology of the liver into clinical applications. Dr. MacParland and her research team will investigate how liver immune dysregulation drives liver diseases, including chronic viral infection and inflammatory liver fibrosis. They will also examine how the liver immune environment can be therapeutically manipulated using nanoparticles to slow or reverse ongoing damage. Dr. MacParland will be telling us today about her work in developing a cellular map of the liver and how this may be used, hopefully, to better understand PSC. Welcome.
Thank you, Miriam. So I'm really glad to be here and to meet more of the PSC community. I've met you at conferences, at fundraising events, and your enthusiasm and your eagerness to partner with us as we try and tackle this terrible disease is absolutely fantastic. So I'll give you a little bit of background on myself. So I come from Newfoundland. I did my undergrad there. I did my master's in Birmingham, where Gideon worked for a while, and we brought him back. And then I went to Germany. I did a PhD in Newfoundland. And then I came to Toronto and was recruited to the UHN. And so, traveling around the world, it was really stunning to see how the Toronto area, the Toronto liver community, they really work together, and how the UHN, the Toronto Centre for Liver Disease, and the transplant program, they're very intertwined. And we're part of a global, or a group kind of drive to taking basic science discoveries and driving them towards these therapies that Gideon was talking about. And then through the research center, some of you will recognize some of these researchers, and these are the basic science researchers in the research center, and of course the clinical researchers that you also see in the clinics are also a huge part of this. But you can see there's people of various diverse backgrounds, and one thing that Gideon mentioned, and I really want to reiterate, is that research, cures, innovation, it's a team effort, and it involves people of various backgrounds, including patients, including community advocates, and kind of together we can really tackle disease. And so, as Gideon mentioned, we all know that the liver is required for digestion, for blood clotting, for clearing toxins and fighting infections and the regenerative processes. But as scientists and immunologists, we really don't know the cells in the liver that are required to keep all these processes going. So for the most part, the liver is homeostatic, and that means that it's keeping it very low profile. It's very quiet, it's keeping things status quo, but we don't know why. And so as a scientist, you're always told, ask very focused questions. Okay, so that's very important in science. But when you study the liver, there's lots of questions that have absolutely no focus. And the big question that we're addressing, or we're trying to look at, is a not so focused question, and it's what kind of cells are in the liver? And the reason we're asking that question is, in people who have liver disease, how are the cells that are in their liver mediating disease? And so mapping the cellular microenvironment of the liver is required to define and target cellular processes that drive disease. And so the states of the liver that we're thinking about are liver regeneration, because in general, in a happy liver, if you have a liver insult, there's a regenerative process that takes over and restores homeostasis. But then you have situations of disease, including liver cancer, including inflammation, including PSC, and transplant rejection, and it's all kind of part of the sphere of liver disease, in which these cellular processes are off balance. And so there are cells of the immune system, including macrophages, that can really modulate this or can drive this. And so macrophages are kind of the eaters of the immune system. They're picking up things that are going through the liver. And of all the organs in the body, actually the liver holds the most macrophages. So even though we're taking an unbiased look at all the cells in the liver, we're really watching the macrophages because we know from histology and from lots of studies that have been done before that there are a lot of macrophages in the liver. And for example, macrophages like immunoregulatory macrophages, so macrophages that are inappropriately quieting the immune system, come to play in situations like liver cancer, where you want to have cells that say, hey, that's a cancer cell, that should be deleted and removed. In that situation, the macrophages are actually inappropriately quieting the immune system. In the case of PSC and inflammation, there are cells that are inappropriately activating the immune system, and they're calling in troops when they really shouldn't be calling them in. So what we want to do is understand what cells are driving those processes so we can target them specifically. And so I'm going to describe a study that we embarked on to bring us to the point that we can start to study disease. And the rationale behind looking at the liver with this type of technology is that our current understanding, or our understanding at the point of initiating the study of the biology of the liver was not as defined as we needed it to be in order to find specific targets to modify to treat disease. And so what we thought about the liver was we would have biopsies, we would take those biopsies, we would look at the overwhelming signal in those biopsies. So we're kind of looking at the liver almost like a smoothie, so that, whatever the average response is, or the average, for example, cellular signal is in the liver, well, that's the signature of disease. And so, but what we know is that there's distinct populations within the liver, and their frequency within the liver can have a play, or can be related to disease, and that these cells are responding to environmental cues based on what's happening in the liver. And what we know is that cell signatures within the liver are associated with poor or good responses to treatments, for example, like antiviral therapy. But really, in order for us to take it to the next step and to understand which exact cells to target for each disease, we really need a clear understanding of the roles of each of the individual populations within the liver. So we need to kind of go from this smoothie to the individual fruits that are making up the smoothie, and how many of those fruits are needed for a homeostatic or a healthy liver, and then what's happening in a diseased liver. How is that fruit bowl messed up? And so the study that we looked at was a study to look at the complex structure of the liver, and that's possible using something called single-cell RNA sequencing. So the main objective of this study was to create a comprehensive cellular map of the liver and to use that as the foundation for understanding disease within the liver. And so one of the most important parts of this study was the way we actually handled this tissue. And so the acquisition, we actually acquired tissue from parts of transplants that were appropriate, or livers that were appropriate for transplantation. We cannulated or put needles into that liver, and then we flushed out anything that was circulating through that liver, because we really just want to look at the cells that are in the liver that have been recruited in and that are mediating either health or disease. So then we add little tiny enzymes into that liver, and then we can take all those cells apart. And so we try and do that as gently as possible. And then we open up a tissue layer that has been holding those cells together, and those cells pour out. And so then what we can do is we can take each of those cells and we can put it through a cytometer where it incorporates into a bead and an oil droplet. And the details of what happens there aren't as important as the fact that every individual cell, you can see what they are and how they're signaling. And so then you can kind of say, well, how many cells? Is it the number of cells in the liver, or is it what they're individually doing that's important in health and disease? And so then we go from the individual populations in the liver, and then we use our collaborations with our bioinformatics colleagues to take all those cells and then we cluster them. So what we see is these individual populations are your map of a healthy liver. And you'll see, if there's too many bananas, is this one disease? If there's too many oranges, is this another disease? So this is called single-cell RNA sequencing. So what we did in a huge collaboration with the whole kind of Toronto liver community is that we looked at 5 healthy livers. And so these are lobes taken from livers that were acceptable for transplantation, but that piece of tissue was normally discarded. So we took that and we took it apart. And what's nice is we can actually look at how that liver did in that patient as well. So we can follow that liver the whole way through. And so what we saw was previously, it was very well known that there were lots of hepatocytes in the liver and that they're zonated and that they have different kind of roles depending on where they are in the liver. But what we were able to show, we could actually identify the cholangiocyte populations, but I want you to remember the fact for the next talk that those cholangiocytes were a very small population, whereas all of you know that we have a lot more cholangiocytes than the smallest population in the liver. We saw cells called plasma cells. They secrete antibodies and B cells, and mature B cells. And what's important is these are cells that are mediating immune responses. So we're starting to see that the liver is not just full of hepatocytes and cholangiocytes, but also a lot of immune cells, even in a healthy liver. And we saw lots of T cells and NK cells, and NK cells and T cells can respond to antigens, but they can also mediate inflammation. And some of these T cells can actually dampen immune responses. And then we saw different types of endothelial cells. And what we can do, because we can understand the cells at a single cell level, is then we can take some of the signatures from these cells, and then we can look at whether the zonation or the way these cells are represented within the liver is dysregulated. So it's another way to look at whether or how the disease establishes itself and the possible targets that we can look at to modulate disease. And then what we were really interested, again, were the eaters of the immune system, those cells that, they sense everything that's coming from the gut that Gideon was talking about, but normally they clear those antigens and they don't activate, the recruitment of all these different immune cells to mediate inflammation. They kind of do their job and they say nothing's going on. And so those are non-inflammatory macrophages. So they're secreting factors that mediate regeneration. So they're causing proliferation of hepatocytes. But then inflammatory macrophages, they kind of secrete alarm signals that recruit in all these inflammatory cells. And so what we saw was that there's two different populations of macrophages, even in a healthy liver. And so it allows us to kind of speculate that we could look at those populations and their role in disease. And you don't need to look at, you don't need to know these genes, but the reason that we know that they're non-inflammatory, because again, this is an unbiased approach, is that the key genes, if you look at what those key genes do, they're mediating inflammation. And if you look at the key genes in the non-inflammatory cells, they're quieting the immune system. But what we want to do, and again, the power of looking at a liver on a single cell level, is you can actually not just look at what their identity is by what genes they're expressing, but you can actually see what they're doing. So if a cell looks like an inflammatory macrophage, but it can't secrete those factors I was talking about that recruit things in, then probably it's not an inflammatory macrophage. So what we're able to do is take those cells and we can put them in a dish and we can stimulate them with things that your liver would normally see. For example, bacteria byproducts. And what we can see is that those non-inflammatory macrophages are secreting less TNF alpha than compared to the inflammatory macrophages. So we can see that these two populations exist. We can look at them by single-cell RNA-seq, and then we can look at their function in the lab. And so this is all very important because, for example, in PSC, if we find that these macrophage populations are key players, then we have to have a way to read whether we've modified them with a drug that we've developed based on the single-cell RNA sequencing. So functional assays are really important. And then I wanted to show you some of this bioinformatics, because the important thing, or the very neat thing that I've learned over the past 2 years, is that there are a lot of brilliant people that can take your data and do things with them that you never even knew could happen. And so Gary Bader at U of T, he can take this single cell data and then do this cool map called a pathway analysis. And what it can say is, these cells, because of these key genes they're expressing, this is what they're doing, and this is what they're doing, and this is what they're doing differently. And what that means is that you can kind of either confirm what you think those cells are, but also, if you can see what's active in those cells, then you can use something called drug discovery pathways, where they can come in and repurpose drugs that were maybe developed for thyroid disease. And then you can see that actually, you know what, based on these maps that are unique to the patients, you could use the thyroid disease to treat PSC. So you repurpose drugs that are already available. So that's kind of the dream. But for the purpose of this study, we used the pathway analysis to confirm that in fact these inflammatory macrophages had active pathways in bacterial defense and innate immune responses, whereas the non-inflammatory macrophages, so these regenerative and quieting macrophages, had active pathways in suppression of cytokines and inhibition of myeloid cell or myeloid differentiation. So what that brings us to is a model where we can start to delineate what cells are in the liver, where they're found in the liver in a healthy individual, so we can have all kinds of guides to look at the diseased tissue. And you know, so we developed this map, and it was actually the world's first map of the healthy liver, and we can use that to kind of as a gold standard to compare a healthy and diseased liver. And so right now we're expanding the map, and we've included livers from across multiple donors. And one thing I'll talk about a little later when I talk about our future research is we're trying to expand this to pediatrics and all the way through, you know, to adults, because we know liver disease, particularly PSC, particularly fatty liver disease, it's different in children than adults. And then what we really want to do is make an impact. So as Gideon very well outlined, science has changed. We need to use all the brains to make discoveries. And so we're sharing all this data openly, and because we have unique access to this healthy tissue. So we're actually taking the data, we're sharing it through the Human Cell Atlas Consortium. So other people that, for example, may have, you know, transplant rejection cohort, but they don't have a healthy cohort, they can use this as their comparator. So we can have a bigger impact than just on the diseases that we're focusing on at the moment. And then the way we translate those results from our healthy map right now is we're looking about how we can, or we're looking to how we can target immune cells to promote regeneration. And then we're actually developing maps of mouse livers, because if we can promote regeneration in mouse livers, then, you know, possibly we could translate that to humans. And then we're looking at whether we can selectively and repolarize immunoregulatory macrophages to promote the detection of tumors in patients with hepatocellular carcinoma. And this is using an animal model of hepatocellular carcinoma, which is kind of neat. It's the woodchuck. So we work on woodchucks. And so we've developed a woodchuck map of the liver. And then, as I'm going to get into this after the lunch break, is we're really interested in understanding the immunology and the cellular biology of PSC, so that we can find new targets to treat and prevent the need for transplantation. And it's funny because I'm actually a member of the transplant program, but my job is to put them out of business. So. Yeah. So just in summary, this is, you know, I wanted to kind of show you our recent map, and this map is, you know, we're looking to describe and bring a new light into our understanding of the cellular biology of the liver. So we're able to look at 5 healthy livers. We had looked at 8,444 cells. We contributed that data to public domains, and that's actually been uptaken quite well. And this is our foundation for our disease studies that I'll talk about this afternoon. And of course, I'm just a representative of the team, and this team is a huge team, and it's constantly expanding as we delve into different diseases. And so really, none of this is possible without every member of this team, and everyone is very, very driven and very excited about the possibilities, especially within the liver community in Toronto. And I'm happy to take questions.
So we have about